Other questions for Sam?
Oh, yes.
So Molly Schweik at U of T. Do you have any issue with solubility?
No.
No, none at all?
These are wonderfully soluble in water and polar organic solvents, and as we put more greasy groups on them, they become more and more soluble in non-polar solvents. I think it's because they have their rigid structure breaks up their hydrophobic patches and their hydrophilic patches, and they're frustrated because they can't adopt different conformations to become self-complementary. There's only been one case where we tried to make molecules that could hydrogen bond with each other. That stuff was brick dust. Everything else is very soluble in water, water acetonitrile, chloroform, DMF, all sorts of solvents.
Really nice talk. Really
Thanks.
interesting. Jay Hickman, UCF. Have you tried to apply the software technology you're using to design these things to look at A beta and look and see how A beta forms oligomers?
A beta is you mean?
Amyloid beta, it's the peptide in Alzheimer's disease.
So the software that I have is what it's doing is it's taking these rigid building blocks and then connecting them together, applying the matrix math to figure out where building block I+1 attaches to I. It can do this with amino acids, but amino acids are so conformationally flexible that you have this enormous conformational space that you need to search, and it becomes a protein folding problem, which my software could tackle, but it's really focused on what I tried to do here was design monomers that are more computationally tractable than amino acids. And the software can do amino acids and sugars and anything else, but we're tackling the bispeptides.
Very nice talk. Shui Li from MCG. I have a quick question. One of your slides showed that you labeled your peptide with fluorescent for the labeling. My question is, after you label this fluorescence, do you saw any interference with your protein or peptide function?
Actually, we think it's involved in binding the protein because there was one stereocenter that we had set one way initially to lift it away from the protein if those three groups are binding into the pocket. And then on a LARC, Zach flipped that stereo center, which would push it back into the protein. The binding improved. So there's hydrophobic surface up there to make contact with, and we think that the fluorescein might be contributing to the binding now. Okay. And we're just now taking it out and looking at it in competition experiments to see how much.
The reason I'm asking is that we are trying to monitor the protein trafficking through the membrane or to the nucleus. We found that once we label something with fluorophore, it will interfere with the behavior of protein.
Yeah.
Okay, thank you.
So, Stephen Lee, OSU. I wanted to mention something that you mentioned really only in passing in your talk, but it's potentially very important, and that is this is one of several nanoscale antibiotic materials I've seen people propose, and most of them work by somehow interfering with membrane function. And the thing that's potentially important about them is if you go back and study the literature of plasmid biology and you look at where antibiotic resistances come from, they mostly come from genes that were present in the synthesizing organism that subsequently end up on an X chromosomal element and are transmitted horizontally. Right? Whereas these sorts of compounds have the advantage in that there is no synthesizing organism except you. And so therefore the possibility is that resistance to these things may be much slower kinetically to develop in the population than conventional antibiotics. And this is important because I think we're in the process of returning to the pre-antibiotic era. And so infectious disease was a major cause of morbidity and mortality in the past. Having new antibiotics certainly would help us to avoid going back to that state.
Yeah. Well, our bodies right now make antibiotics like that. They're called antimicrobial, cationic
like defensins
antimicrobial.
and meganins.
Yep. And bacteria have not evolved much of a resistance over the millennia. The feature that those peptides are exploiting is that bacterial and membranes are primarily negatively charged on the outside, while human cells and mammalian cells are neutral on the outside. So cationic peptides electrostatically stick to these membranes. And, you know, we're making them all the time. They're protecting us. They're part of our ancient immune system. Insects don't have T cells and antibodies. They have primarily
Fungus and
antimicrobial
things like
peptides,
that.
and they do very well with them. What we're trying, the problem with them is that you need a fairly high concentration because they have to self-assemble, and they're only effective, you know, the bottom is maybe about 1 micromolar to 10 micromolar. So they'd be very expensive to use they're very expensive soaps basically. But if we can pre-assemble a sort of a portable whole that could drop into a membrane and not have to self-assemble, they could work at very, very low concentrations, and that's why it's sort of a nanotech approach to antibiotic.
The point I was making is T1 have to go from introduction of a new antibiotic to prevalence of resistance is now like 10 to 15 years. It's not very long. So the functional life of the antibiotics are limited. There's a rational, reasonable reason to hope that things like what you're suggesting might actually have a longer, useful clinical life.
I hope so. Thank you.
Final question?
Yes, it's not really a question, it's more like an idea. I don't know if you've ever heard of the work of the Demain, Eric and Martin Demain. There are two MIT professors, and they work on origami, molecular origami. And there is a lot of work done with DNA, which has a lot of problems. Well, your stuff, especially the one that you were showing at the beginning with the molecular hinges, it's something that really recalled me of that work. Have you ever considered to do anything in that direction? Because there are a lot of applications in the nano world for molecular origami.
Actually, I think I know how to make protein-like objects and complex asymmetric materials, because we can put metal binding groups on these things and do metal templated assembly. We're starting to do that now to build three-dimensional structures. We can also make short oligomers and then chain them together like building blocks. So we can make protein-like structures that would be kind of like segment, slats connected by hinges with appropriate functional groups so that they could fold up, but like a child's puzzle, as with lots of complementarity on each subunit. And we're working on that too. It's synthetically, I totally see how to do it now, and I've got people working on it. Sure.
What do you mean childlike puzzle? Ones that are...
Well, a kid's puzzle has big pieces,
Right.
and lots of it's obvious how they fit together. A adult's puzzle has thousands of pieces and they're tiny. I think the protein is like a adult's puzzle, and what I'm trying to build is like a child's puzzle, easier to put together. My 5-year-old would appreciate that.
He'll be helping